Hello everyone and welcome to our WGCNA workshop, and I congratulate you on Christmas, and also I hope you have a nice year. And let me to share my slides for you and start the my slides is visible for you or not, please say me. Okay.
Visible.
Okay, thank you so much. Today we're gonna talk about the WGCNA package. It's a node package for weighted correlation network analysis. WGCNA, one of the useful and user-friendly package based on R, but it has some details, and while using it, it's better to pay attention to some biological and statistical points. And today I'm gonna talk about these important. This is one of my last papers, and we could publish it in Frontiers on Genetic journal in 2023. Based on this paper, we could identify the differential expression genes related with heat tolerance in chicken. And for invalidation part, we use the WGCNA analysis tool. Therefore, all of the details we were used in this paper, we're gonna talk about all of that. These are my presentation titles. At first step, we're gonna talk about the why network. Why it's better to using the network instead of the list of genes, individual list of genes. And how to construct a weighted gene co-expression network, and what types of data are suitable for WGCNA as an input data. And association with other external traits, and we're gonna talk about two. And after that, in each modules there are some hub genes or one hub genes. We're gonna talk about the hub genes or key genes. What's that, and how can I get the hub genes or key genes? And then we're gonna get the list of genes in each modules. And these list of genes are really user-friendly, useful for us for our downstream analysis. For example, we can use these list of genes as an input data for cytoescape gene ontology or string website or others. And at first step, we know there are different methods for identifying the differential expression genes. For example, we can use the COFDIF, DSEC, and EDGR for getting the differential expression genes based on the p-value. And we said that if each gene have less than 0.05 or 0.01 p-value, yeah, that's the significant gene. But all of these results, these three software results, aren't overlapping together and aren't same, and there are a difference between these results. And on the other hand, the statistical significance aren't completely equal with the biological relevance. Therefore, after identifying the significant genes, we have to some validation analyzers. One of the important validation analyzers is using the WGCNA package. And we know the complex rate controlling with multiple genes, or it's better I said that, the complex rate controlling with multiple groups of genes. And it's better to identify that groups of genes instead of the list of genes, because in this format we calculate the correlation coefficient between these genes, and these genes have more than 10 zero between that data, we're gonna get a high positive correlation between that genes, or it's better to say we're gonna get the significant positive correlation. But that's false positive, and it's better to removing unexpressed gene. And also, WGCNA doesn't recommend filtering genes by differential expression. And I said that, for example, in chicken we have 28,000 transcripts, and in our study, we should get just 200 of that are significant. And maybe I get that it's better to using just these 200 genes instead of that 28,000. But that's strongly wrong, because by filtering genes by differential expression results in a set of correlated genes that essentially form a single or several highly correlated model. And also, if we do this filtration, our data can't be fit on scale-free topology model. And about the scale-free topology model, we're gonna talk completely in next slides. That's one of the important parts of the WGCNA package. And then, after that, we have to some data pre-processing. And it's better to remove the missing value. For example, in this figure, this figure shows our sample, and the rows is our gene name, and the columns show us the sample. For example, this gene have 3 missing value. Therefore, if our gene have more than 3 missing value, it's better to removing that. And then we can, by good sample gene, remove all these genes. And on the other hand, about the outliers, sometimes we have a special sample, for example this sample. All of the genes in this sample have higher gene counts than the other samples. Therefore, we said that this outlier, and it's better to removing that. For identifying the outliers, we're gonna use the HC classed function in R. And let me to show you in our environment how can you get the outliers and missing data. I'm using the Linux system, but there aren't any difference between the other systems, and you can use the R or RStudio in Linux, Mac, or Windows, and don't have any difference between that. I'm running the RStudio, and after running of that, we can RStudio. This is loading. I installed the R or RStudio based on the conda codes in R. Therefore, I use these codes for running of that. Okay. And also my codes here are ready for running. Okay, at first step we have to installation of the WGCNA package. We know the WGCNA are based on Bioc Manager, and therefore for installing of that we have to use a Bioc Manager install code. And therefore, if you don't install the BIOC manager, you can install that by the install package BIOC manager code. And then you can load it by library BIOC manager. And after installing the WGCNA, you can load it with the library WGCNA. I installed that before, therefore today I don't want to repeat it. This is running the loading required package, and okay, that loads completely and we don't get any error or warning. And then in this part we have to introduce our directions for the software, and all of my input data are in this direction in desktop and WGCNA folder. And based on the get WD, we can check my code is okay or not. Yeah, absolutely, that works completely. And therefore, I want to load my data, my VST data. Let me to get the head of the data and show you. Okay, and these are in the row names or our gene names. And also all of the columns show us the sample names. And by the dimension code, we can get the data's dimension. And therefore today we have the 1499 gene. Amounts of data, and no, these count by just keep these 31 samples for me and removing this one. Preparing our data, we have three types of method, and one of that is automatic method. It's one-step network construction. It's not suitable method and it's not flexible. But step-by-step condition of gene network and identification of module is one of the suitable method, and today we're gonna talk about all of the steps of this method. And about the black-wise method, if you have a huge amount of samples, therefore it's better to using the black-wise network, because based on this method, all of your samples be divided in different parts, and the WGCNA try to analyze that in parallel. And therefore, if you have the huge number of the samples, it's better to using the blockwise. And in the step-by-step sample, we're gonna calculate the correlation matrix, agency matrix, a topological overlap matrix, and completely we're gonna talk about in next slides. At first step, we have to choose the correlation method. There are three correlation methods. For example, one of that is Pearson correlation method, and we can calculate the Pearson correlation method with a core function in R. This method is suitable for continuous data, and also the gene counts are continuous. Therefore, it's one of the suitable methods for correlation for WGCNA package. And based on Pearson, we can measure of the linear correlation. It is the fastest method but sensitive to outliers. But we talk about the outliers. Don't worry about that, and we can identify the outliers and removing that without a doubt. And also the other one is Spearman method. And Spearman method is suitable for binary data or categorical data, and it is rank based and less sensitive to gene expression difference. Therefore, it is not suitable for WGCNA package. And also the other one is bi-weight. Of the genes in unweighted network. please attention this part. If we just calculate the correlation between the gene I, gene J, it's equal to 0.8, and also I correlated the correlation between these two genes and it's equal 0.2. But the fold difference between these two numbers is low, it's equal 4. And on the other hand, if we use the power equal 4, I get these numbers. The agency element is equal to 0.4 and the agency element between gene M and gene N is equal to 0.001. Therefore, there are a high fold difference between these two correlations or agency elements, and this is equal to 256, and absolutely we can say this is between these two genes there are a strong correlation, and on the other hand I can say there are a weak correlation between these two genes, between M and N there are weak correlations. For choosing the strong correlation and weak correlation with high accuracy, we have to use the power. But how can I choose the best amount for power? For example, we can use the power 4, 6, 9 or 10 or other numbers. Which one is suitable for us? Okay, by pixel threshold in WGCNA package, we can set which powers are suitable for us. In this plot, in horizontal part we have the soft threshold amount, in vertical part we have the scale-free topology model fit. Step by step, this package use all of these numbers. For example, at first step use the number 1, then we use the number 2, then number 3 instead of this power in this formula. Then get the data and the new elements, and after that fit it on topology overlap model, and based on each these numbers we can get the higher and each of these powers are above the R squared more than 0.9. We can say this power is suitable for us. And let me show you how can I get the powers based on the WGCNA package. Okay, for example, I choose that I want to try the agency matrix and calculate the agency matrix for these 10 numbers, 1 to 10. And after that, by pixel threshold, I'm gonna get which one is suitable for me. It's better to get the, okay. All of that calculate the R squared amount for me. For example, if my power is equal 1, the R squared is equal to 0.1. And if the power is equal 6, the R squared is equal 0.899, and this is suitable amount, and this amount is so near to 0.9, and it's the first number or under the R square red line. For getting that plot, I can use this formula. Okay, I get all of these data's plot. In this part we have the soft threshold 1 to 10, and in this part I have the R squared amount, and we said that each number are above the red line or R squared equal to 0.9. That's my suitable power. And in this example the power is 6, and we used we're gonna use the 6 as a power in next steps. And also I want to talk about the scale-free topology more. What's important for us. We know most of the traits in biological are complex traits and controlling with multiple genes. And let me to check my cell phone, and I have a, oh no, no, don't important one. Okay. We have some complex traits, and these complex traits controlling with multiple genes, and or it's better to say that with multiple groups of genes. And I mentioned that in chicken, for example, we have near 28,000 transcripts. But all of that don't have any significant role with our special trait or our special condition. And most of that don't have any interaction or link with together. And most of that are in this part of the plot. In this part of the plot, we have lots of genes or transcripts and don't have any interaction or link with together. And on the other hand, we have some genes as a significant genes and high the low connectivity with together. And so, therefore, and this distribution name is scale-free topology. And that beta or power help us to get data or elements are fitted on these plot. And in this plot, this part or horizontal part show us the connectivity number, and the vertical part show us the frequency. And what's the connectivity, and how can I calculate the connectivity? This is the connectivity formula, and I have an example in next slide for calculating that. For example, after calculating the agency matrix, I get this matrix, and all of elements are getted with the agency method. Function, we can calculate the And based on the hierarchical clustering, and we're gonna hierarchical clustering of the this term amount, and after that we're gonna get this plot. In this plot we didn't choose the minimum size and we didn't use the colors. And in the next step we said that our minimum module size be in 30, and please use the colors for our plot. Okay, based on this plot we get the different types of the modules, for example brown, blue, yellow or green modules. Please attention in this part we could identify the 5 module: blue, brown, green, turquoise and yellow. The gray part isn't important for us. The gray show us a genes. Some genes haven't don't have any connectivity with together and aren't in subset of our modules. And therefore we have 5 modules based on this analysis: blue, brown, green, turquoise and yellow. And all of the unconnected genes show with gray color with us. And after identifying the modules, we want to know, are there any interaction between each of these modules with our external trait? For example, our external trait is blood pressure or insulin or production trait in animal, in plants or other traits. And in this example I use the binary form of the data, and because we had 2 types of group we had some chickens, and some of that be under the heat stress condition and some of that be in control condition, and we could get the association or correlation between these conditions and these modules. For example, in this study we could get the near 23 modules, and I want to know which one of these 23 modules have significant connectivity or association with our trait. Let me to explore that. Please attention in module black. In module black we have 2 numbers, and one of that is minus 0.24. This shows us the number of the correlation coefficient between the eigengenes of this module and head stress amount, and all of the numbers in parentheses are our p-values, and we know each p-values less than 0.01 and 0.05, we said that that's significant for us. And so for example in module black this is not significant because the p-value are equal to 0.2. And in module green we get a high negative correlation. It's minus 0.75, and this is shows significance because is less than 0.01. And therefore step by step we're gonna check all of these parts and choose just significant modules, and because the significant modules are important for us, and after that we're gonna get the list of genes for these significant modules, and the others aren't important for our analysis. And let me say we can use different types of the trait as an input data for WGCNA. For example, sometimes our data are in binary format like this, and sometimes we have categorical data, or sometimes we have the continuous data, so, maybe height, weight, blood pressure or others. And also we can mix all of that and get in a plot like this, and don't have any limitation about data form. And let me to calculate for you in R and getting this plot for you. Okay, in this part, this is my external trait in condition, in different condition. For example, I had two condition, heat stress and control. This is my external trait. Let me show you, but get in the head. This is binary format, and I had just 1 and 0 amount for that. Based on this formula, we can calculate the eigengenes for each module, and after calculating the eigengenes for each module, we're gonna calculate the correlation coefficient between the eigengenes and our data. Based on this code, it's the correlation between the eigengenes and our data. And after calculating the correlation, I want to get the p-value amount too. And let me to make this plot for you too. Okay. Based on this plot, we can set which modules are significant for our trait. For example, in this figure, the module green and the module blue turquoise are significant for our trait and our condition in heat stress. For example, and after that, I said that it's important for us to know which genes are in our significant modules. For example, one of the significant module name is green, and by this code we can get the list of genes are in module green, and all of these genes are in module green. And also you can choose the another two, and don't worry about that. For example, you can choose the another color names, maybe yellow or others. Okay, everyone, and the resource I use for talking about this workshop. And also we have another workshop. It's extended and completely practical workshop, and this is our next workshop poster. Therefore, if you are interested to competition in our next workshop, please join this link, and this is the Telegram groups of link. And in our next workshop, after registration of that, we're gonna send you all of the codes and an example data, and step by step you're gonna run all of the codes with me. And if you get any problem and any error, we're gonna solve it step by step. And also we have some points about the pre-processing of data. How can I normalizing of data instead of how can I normalizing of data before using that as an input data for WGCNA, and how can I get the hub genes, and how can I get the interaction networks between modules and heat map plot, and there are some other points about the WGCNA package. We're gonna talk about that completely, and after that you can run completely your data with WGCNA package. And also, if you have the bulk RNA-seq data, single cell RNA-seq data, or if you have the methylation data, protein count data, you can use this package, and you're gonna get the suitable results for your study. And our next workshop is on the 13th and 14th of January. And if are you interested to joining us, please join this group, this Telegram group. And after that, we're gonna send you the link form registration. And this is our email addresses. If you have any questions, I'm ready to answer that. And at the end, I'm really thankful for your attention. And if you have any question, I'm ready to answer your questions.
Okay, thank you, Dr. Hussein, for this informative and engaging presentation. Please answer the questions in the chat box, and if anyone have any question, raise your hand.
Yes, you can use it for single cell data too. There aren't any difference between the single cell data and bulk data. It depends on your aim. And sometimes we want to identify the co-expressions, or sometimes we want to identify the interaction between the between RNAs. Yeah, you can use the WGCNA package too. If you have any question, I'm ready to answer that. Okay, we're gonna share the WGCNA group in chat bar too. Let me to copy and paste that. Please paste the WGCNA group link in this part. And I can see your question. What's input data for WGCNA from RNA-seq data analysis? Gene counts, normalized gene counts, FPKM.